Module 5, Cell Signaling, Part 1.
Up until now, we have been talking about cells as individual entities, but in reality, cells don't really exist alone or by themselves. In a multicellular organism, survival depends on the ability of cells to communicate with one another about growth, differentiation, and metabolism. And collectively, this is known as cell signaling. The cells can signal one another or communicate with one another through a number of different cell signaling molecules. There are literally thousands of different signaling molecules, and at the bottom of the slide, you will see a list of general classes of signaling molecules, but this is a very abbreviated list, and each of these components have many entities within them. So there are literally thousands of signal molecules in the cell. Now, in some cases, a signaling cell will release a signaling molecule through a secretion or exocytosis. And this signaling molecule can then be received at a part of the target cell through a receptor. This is probably the most common form of cell signaling. In some cases, the signaling molecule itself can remain very tightly bound to the plasma membrane of the signaling cell, and it can even be a transmembrane protein as shown in the slide. In this case, the communication with the target cell occurs by physical interaction between the two cells, but this is a much rarer form of signaling. In this discussion, we will focus on secreted signaling molecules that you saw on the previous slide. Now, regardless whether the signaling molecule is secreted from a signaling cell or whether it is a part of the plasma membrane of the signaling cell, the message or the communication is received by the target cell through either receptors or gap junctions. Now, if you think about secreted signaling molecules, you can divide them into two broad classes, and we're going to talk about them this way. They can be hydrophilic signaling molecules or hydrophobic signaling molecules. In the case of hydrophilic signaling molecules, seen on the left side of your slide, this often interacts with the target membrane through cell surface receptors. So rarely does a hydrophilic signaling molecule enter the cell to propagate the signal. Now, on the other hand, the hydrophobic signaling molecules, which are seen on the right-hand side of the slide, use intracellular receptors, so it's necessary for these signaling molecules to pass through the plasma membrane in order to encounter their receptors. In the example on the slide, you can see a hydrophobic signaling molecule being delivered to a target cell through a carrier protein. And this makes sense because if it's hydrophobic, it needs to have some kind of carrier protein to shield it from the aqueous environment of the extracellular space. At the target cell, the hydrophobic signaling molecule passes through the plasma membrane and then binds to an intercellular receptor. The other way in which signaling molecules can be passed from one cell to the other, or another way cells communicate with one another, is through the gap junctions. And these are relatively large structures that are formed between the plasma membrane of two opposing cells, and they contain a small pore through which very small signaling molecules can pass through. We are not going to consider gap junctions anymore in our discussion of cell signaling, but when we talk about cell-to-cell interactions in another topic, we will revisit gap junction. Whether a cell will respond to a signaling molecule and the way in which it responds to a signaling molecule depends on a couple of things. First, it must have a specific receptor that can bind the signaling molecule. How the cell responds to signaling depends on the intercellular signaling machinery that it has. And we are going to look at some examples of signaling machinery. And because of this, different cells can respond in different ways to the same signaling molecule. So for example, on this slide, in a diagram of signaling by the signaling molecule acetylcholine, which is a neurotransmitter. And you can see acetylcholine can induce contraction in the skeletal muscles. It can induce relaxation in the heart muscle, and it can induce signaling in the form of secretion in some secretory cells. So it is very interesting that the same signaling molecule can induce different responses in different cells. And again, you have to remember that this is due to the intracellular signaling machinery that is present in these cells. Now, there is one or more general comment that I want to make about signaling before we start looking at some specific examples, and that is that there are 4 forms of signaling mediated by secreted signaling molecules, and these forms of signaling can either work in a very short distance or in a very long distance. So for an example, here is a signaling releasing signaling molecules shown in the red, and in this case, the signaling molecule is acting as a local mediator and only influencing the target cells in the immediate vicinity. This is known as the short-range signaling or paracrine signaling. But in multicellular organisms, short-range signaling is not sufficient. So multicellular organisms have evolved to have some very interesting and very sophisticated long-range signaling pathways. So the synaptic signaling shown here is probably one of the most sophisticated types of long-range signaling. In this case, a neurotransmitter is released from the axon of the nerve cell and receives by a target cell. This is known as long-range signaling because the axons of some nerve cells can be very long, up to 1 meter in length. Now, endocrine signaling is another long-range signaling, which is shown in the bottom of your slide. And in this case, an endocrine cell will release a hormone that can enter into the bloodstream. It can exit the bloodstream at much farther distance, and it can influence the target cells at a much longer distance from its origin. Now, those 3 methods of signaling all involved a signaling cell communicating with a target cell that was different from the signaling cell. So signaling was between 2 cell types. However, the signaling cells communicate with the same cells or even themselves, and this is known as the autocrine signaling. And this is another form of short-range signaling. So here is an example in the slide. You have a number of cells of a signal cell type, all releasing signaling molecules and communicating with each other. And in some cases, those signaling molecules are received or bound by the receptors on the actual cell that released a signaling molecule. Okay, now, what I want to do is look at some specific examples of signaling molecule and signaling machinery. And we are going to start out with the hydrophobic signaling molecule because they are very easy to discuss because they all function in the same way. So you can talk about the hydrophobic signaling molecule story as a group and in very general terms because it applies to all of them. So the hydrophobic signaling molecules can be divided into 3 groups. They are steroids, the thyroid hormones, and the retinoids. On this slide, you will see the steroid hormones, and these are just a few examples. There is cortisol, which is released by the adrenal gland, which sits on top of the kidney, and this signaling molecules can influence metabolism in many cell types. There is estradiol, which is also known as estrogen, and then there is a testosterone, which are produced by the cells in the ovary and the testis respectively. They can regulate the development and the formation of secondary sex characteristic. And vitamin D is also considered a steroid hormone, and it plays an important role in calcium regulation, and it is actually synthesized on the skin cells in the presence of light and then further modified in the liver as well as the kidney. Now, you will notice that all of these steroid hormones all have a very similar planar structure, and this is because they are all synthesized from the cholesterol. So these are all your hydrophobic signaling molecules. The thyroid hormone are the second class of hydrophobic signaling molecule, and the thyroid hormone are often made from tyrosine. And you can see here for this example, the steroid hormone thyroxine, you can see the tyrosine within the structure of that hormone with the attached iodines, and the thyroid hormones increase the metabolism in many cell types. And then finally, there are retinoids. And in this example, the retinoic acid. And this is a very important signaling molecule and plays a role in vertebrate development. So the hydrophobic signaling molecules, all of the intracellular receptor have a very similar structure. They are all proteins or protein receptors that have three domains. One domain is known as the activation domain, that is shown here. The second domain is shown here on the right, and then there is a ligand binding domain in which the hormone or the hydrophobic signal molecule bind. And then there is a DNA binding domain in the center. All of these receptors are found in the cytosol, and they are usually bound to some type of inhibitory protein complex, which is sometimes referred to as a chaperone. So in this case, the inhibitory protein complex is known as the HSP19, which stands for heat shock protein 90 kilodalton molecular weight. And this group of proteins, the inhibitory proteins, will maintain the receptor in the cytosol or maintain the receptor inactive in the cytosol. However, when the receptor encounters a hydrophobic signaling molecule, such as a steroid shown here in the red, the steroid signaling molecule will displace the HSP90, the inhibitory protein. It will bind to the ligand or hormone binding domain, and you will get a change in the conformation of the intracellular receptor. And the DNA binding domain will be exposed when the receptor takes on this new conformation by its association with the steroid hormone. This active conformation then it travels to the nucleus, and this can bind to the DNA as a transcription factor. So the response of the hydrophobic signaling molecules takes place in two steps. The first step is known as the early primary response, and that's shown in this slide. At the top of the slide, you can see a steroid hormone that's interacting with the steroid receptor. Once the steroid hormone or the hydrophobic signaling molecule binds the receptor hormone complex enters the nucleus, and the receptor hormone complex can bind to the DNA, to a specific region of the DNA, and increase the transcription and translation of a few different proteins. And again, this is called the early primary response, and this can take place in the cell in as little as 30 minutes. So then what happens? The early response proteins can do one of the two things. Some of them will act on that negative feedback loop shown here and turn off the transcription of the primary response genes. Other primary response proteins will behave as the transcription factors and will influence or enhance the transcription and translation of secondary response proteins. And so think that you can appreciate that a single hormone can induce, or a single hydrophobic signaling molecule can induce enormous changes in gene expression in the cell. When this happens, the cell can change their behavior. Okay, so that's all I wanted to say about the hydrophobic signaling molecule. They all act in the same way. They all use the same mode of action to enter the cell and induce changes in gene expression. For the rest of our discussion on the cell signaling, we are going to focus on the hydrophilic signaling molecules. And you will remember that I mentioned that hydrophilic signaling molecules are usually received at the target cell by a cell surface receptor, and they rarely enter the cell to propagate their signal. So how do hydrophilic signaling molecules bind to a cell surface receptor and propagate a signal through the cell in such a way that the signal arrives at the nucleus and causes altered gene expression, which may lead to altered behavior? The way in which the cells can propagate a signal from the cell surface to the nucleus is through a known system, which is known as a relay system. And these are a series of proteins that can pass the information on. And it is called a relay system because it is very similar to a relay race, where one protein becomes activated, it then activates a second protein, which activates a third protein, and so on and so on. So like the runners in a relay race, the signal can be propagated from the cell surface to the nucleus. And What is really interesting is that the most of the proteins that are found in the relay system in eukaryotic cell are one of the two types. They are either proteins that can become phosphorylated. So when they are phosphorylated, they are activated. Or they are proteins that can bind to GTP, and these are often referred to as G proteins. And here is what this looks like. So here is a general scheme of these proteins that play a role in signaling and their relay proteins. So on the left-hand side is a protein that can be activated by phosphorylation. So you have an inactive protein and it is not phosphorylated. A single molecule binds to the receptor and the signal enters into the cell. That signaling protein becomes phosphorylated. Once the phosphorylated protein is active, it can propagate the signal to the next protein in that relay. Usually shortly after signaling, the protein is dephosphorylated and that signal is turned off. So it is very important for the cell to be able to tightly regulate the signaling by turning these proteins either on or off very easily. And in the case of a protein that can be activated by phosphorylation and can be deactivated by the dephosphorylation. On the other hand, the right-hand side, what we are seeing is the general schematic of a G protein, which you have seen in the previous topics. So again, at the top of the schematic, you can see the relay protein is bound to GDP. It receives a signal and it exchanges its GDP for GTP. And once it is bound to GTP, it becomes activated and it can pass the signal on to the next cell. The intrinsic GTPase activity of the G protein can hydrolyze the GTP back to GDP and the protein becomes deactivated once again. And so you can see that all these proteins act like molecular switches. They're on when they are phosphorylated or bound to GTP, and they're off when they are not phosphorylated or not bound to GTP, but they are bound to GDP. And you will notice that many of these signaling molecules we are going to look are some of these types. Okay, so now let's take a look at a couple of examples of the cell surface receptors as well as intracellular signaling machinery. Now, this slide shows the 7 different classes of cell surface signaling receptors. For this course, we are not going to consider all of these. We are only going to consider only 2 of these and the signaling machinery from the 2 of these receptors. And one of them is the G protein linked receptors, and the other one is an enzyme linked receptors. So let's start with the G protein linked receptors. The G protein linked receptors are sometimes called G protein coupled receptors and are so named because when the receptor becomes activated, it becomes activated when it binds a ligand. Then it interacts with the G protein, and that's what you can see in the slide right here. So here's a G protein that is localized near the G protein coupled receptor, and the G protein is heterotrimeric. What it means that they have 3 subunits, and we are going to see an example of them in a minute. So what happens is the G protein coupled receptor binds its ligand, and this allows the receptor to interact with the G protein. These cause activation of the G protein, and then this G protein can interact with another protein, an enzyme or an ion channel, and activate it. So in other words, it propagates the signal to the next protein like a relay. So we are going to look at 3 examples of these G proteins. One of them is a G protein coupled receptor that activates the enzyme adenylyl cyclase. The second G protein coupled receptor that inhibits the adenylyl cyclase. Signaling molecule is the same, but the activity is different. And then finally, we are going to talk about a G protein coupled receptor that can activate an enzyme which is known as a phospholipase C. We briefly mentioned about that in somewhere else. So all the G protein coupled receptors or G protein linked receptors have a similar structure, and that is shown in here in these two diagrams. They are all 7-pass transmembrane proteins. They have a very large extracellular domain. Their N-terminus is on the outside, and this is the ligand binding domain. So this is where the signal molecule is going to bind. And then on the cytosolic side of the plasma membrane, you can see the arrows pointing to the domains that are responsible for subsequently interacting with the heterotrimeric G protein. The C3 loop, which is the third cytoplasmic loop, is really important in the specificity. And this loop will specify which G protein will interact with which particular receptor. All right, so let's focus on the G protein coupled receptors that either upregulates or downregulates the activity of adenylyl cyclase. So this is a very general schematic of that, and let's take a look of this a little bit more in detail. So here you have a signaling molecule on the extracellular space, and it binds to the G protein coupled receptor. This is in turn activates the G protein, and depending on what type of G protein it is, some can activate the adenylyl cyclase, and in which you would get an increase in the intracellular level of the cyclic AMP. On the other hand, some of the G protein will inhibit the adenylyl cyclase, and it will decrease the level of cellular cyclic AMP. And cyclic AMP is sometimes referred to as a second messenger. And it is called a second messenger because it can go on and then influence the activity of a wide variety of target proteins. So now this is the biochemistry of the formation and the synthesis of the degradation of cyclic AMP. I think that it is important to visualize this biochemically so that you can understand what is going on in the cell. So the cyclic AMP is made up of from the ATP by the action of adenylyl cyclase, and once it propagates the signal, it can be very rapidly be hydrolyzed by cyclic AMP phosphodiesterase to 5' AMP, which is not an active signaling molecule. So the cell has the capability of really controlling the cell signaling by rapid synthesis and rapid degradation of the cyclic AMP. Now, as I mentioned, some of the heterotrimeric G protein will activate adenylyl cyclase, and these are known as the stimulatory G proteins or GS. So I will refer them as GS proteins. And some of the heterotrimeric G proteins can inhibit the adenylyl cyclase. These are known as inhibitory G proteins, and I will refer to them as GI proteins. Okay, so now let's take a look at how this works. Let's focus on the stimulatory G proteins. So at the top of the figure, you can see the G protein coupled receptor in the light green. There's a signaling molecule. In this case, it is a hormone. You can visualize the stimulatory G protein. It is a heterotrimeric protein, and it contains an alpha subunit, a beta subunit, and a gamma subunit. And you will notice that the alpha subunit also contains a nucleotide binding pocket and a GDP binding pocket. And then on the right-hand side, the enzyme is adenylyl cyclase that is going to synthesize cyclic AMP from ATP. So in the step 1, you have the binding of the signaling molecule to the receptor. This creates a conformational change in the receptor such that it can now interact with a heterotrimeric G protein. That's what is shown in the step 2. The interaction of the heterotrimeric G protein with the receptor causes an exchange of the GTP for GTP. And so now the alpha subunit, which is bound to the GTP instead of GDP, is activated. And when this happens, the sulfa subunit diffuses away from the beta and the gamma subunit. So this signaling pathway continues on the next slide, so that at the very top, you have the alpha subunit that is activated, is bound to the GTP, and at the point that the alpha subunit can move through the membrane and interacts with the adenylyl cyclase. And the interaction of the alpha subunit with adenylyl cyclase activates that enzyme. And the enzyme then converts ATP to the cyclic AMP. Shortly after the activation of the enzyme, the alpha subunit has its own intrinsic GTPase activity and now can hydrolyze the GTP into the GDP. This inactivates the alpha subunit. In fact, it is inactivating itself, and then it reassociates with the gamma and the beta subunit. And you are at the beginning of the cell signaling cycle once again. All right, so one question that you might have is how fast does the alpha subunit dissociate from the beta and the gamma subunit? And we can use a technique that we learned about already in the first part of this course when we were talking about the fluorescence microscopy and imaging cells. And that technique is FRET, or fluorescence resonance energy transfer. And I will remind you that FRET is a very powerful technique, and to carry out FRET, you need two fluorophores. And in this example, you have the CFP, cyan fluorescent protein, and you have YFP, or the yellow fluorescent protein. And what is important in choosing a FRET pair is that the emission wavelength of one of the fluorophore must be the excitation wavelength of the second fluorophore. So in this case, the emission wavelength of the cyan fluorescent protein happens to be the excitation wavelength of the yellow fluorescent protein. Now, if these two fluorophores are far apart in the cell and you excite the cell with the wavelength necessary to excite cyan fluorescent protein, what you will see is the emission of blue light and no emission of yellow light, because this emitted light is too far away from the yellow fluorescent protein to excite it. And this could be considered as no signal. And you have to be within 10 nanometers or 100 angstroms in order to get a signal. On the other hand, if these two fluorophores are very close to one another, light emitted from CFP will actually excite YFP, and then YFP will emit in the yellow range. And so you can use this powerful technique to measure that how fast the alpha subunit dissociates from the beta and the gamma subunit. This is an experiment that was carried out by Peter de Vries at Johns Hopkins University. And what he did is he put a cyan fluorescent protein tag on the alpha subunit, and he put a yellow fluorescent protein tag on the beta gamma subunit. And he hypothesized that in its inactive form, when all three subunits were together, if he used an excitation wavelength of 440 nanometers, which would excite CFP, the emission from CFP would subsequently excite YFP, and he would see yellow light. On the other hand, on the right-hand side of the slide, if the alpha subunit is bound to GTP and had dissociated from the beta and the gamma subunit, then if the light at 440 nanometer was used to excite CFP, you should not see any emission from the YFP. You should only see the emission in the blue range, because YFP is too far away from the CFP to be excited by the emitted light. And so what he did again, he CFP tagged the alpha subunit, he YFP tagged the beta subunit, beta gamma subunit, and he transfected these chimeric genes into some cells. And then he followed the emission of the yellow light after the addition of a signaling molecule, which is a CAMP. Now, in this case, he used the cyclic AMP as a signaling molecule, but don't let this confuse you. It is interesting that cyclic AMP can also be an extracellular signaling molecule, but really you can think of this experiment if you add any kind of signaling molecule and you follow the emission of the yellow light, you should be able to determine when the alpha subunit dissociates from the beta gamma subunit. So on the x-axis, you can see the time in seconds. Y-axis is the relative fluorescence of the yellow light. And you can see at about 10 to 12 seconds, he added the signaling molecule. And in about 10 to 15 seconds later, the emission of the yellow light dropped dramatically. And so you can see that this happened very, very quickly. The alpha subunit dissociates from the beta gamma pair quite quickly within 10 to 15 seconds. All right, there are a wide variety of G protein coupled receptors that are able to interact with stimulatory G proteins to turn on adenylyl cyclase. And in this figure, there are three examples: epinephrine, glucagon, and ACTH, or adrenocorticotropic hormone. All of these bind to the G protein coupled receptors, which activate the GS, the stimulatory G protein. And then that stimulatory G protein, the alpha subunit particular, can activate the adenylyl cyclase, and you get the production of cyclic AMP by this adenylyl cyclase. There are also a wide variety of signaling molecules that are respective receptors that can interact instead of the stimulatory G protein with an inhibitory G protein complex, or GI. And again, in this example of prostaglandin E1 and adenosine are the signaling molecule. So when they bind to the receptor, they activate the inhibitory G protein complex, and the alpha subunit will then interact with the adenylyl cyclase. But instead of activating the adenylyl cyclase, it actually inhibits the activity of the adenylyl cyclase. So then the inhibitory G proteins work in the exact same way. The signaling molecule binds to the receptor interacts with the heterotrimeric G protein, alpha subunit binds to the GTP, diffuses from the beta GABA subunit pair, it interacts with the adenylyl cyclase, but at this point, the adenylyl cyclase is not activated, it is inhibited. So it's exactly the same process, and I'm not showing you a diagram of that, but it is exactly like the signaling process that we just saw. What is really interesting is that some pathogens can take advantage of these signaling pathways. So for example, Vibrio cholera can actually hijack the stimulatory G protein signaling pathway. And what happens in this bacterium produces a toxin which covalently modifies the stimulatory G protein, the alpha subunit of the G protein, and it cannot hydrolyze its GTP to GDP. So it cannot turn itself off, and therefore it remains in the GTP bound state or perpetually on. On the other hand, the pertussis toxin, which is related to another pathogen, Bordetella pertussis, which is a causative agent of the whooping cough, can covalently modify the alpha subunit of the GI protein complex, and in this case, it locks the alpha subunit in the GDP bound state or permanently off. And we are going to revisit these two toxins and these two pathogens, I believe, in the second part of the cell signaling discussion. So we are going to learn why these pathogens do this. Okay, so now you have all these G protein coupled receptors on the cell surface, and you have some stimulatory G protein in the cell, and you have some inhibitory G protein in the cell. How does the receptor know which G protein to bind to, which heterotrimeric G protein to interact with? And as I mentioned earlier in this discussion, it is the C3 loop, it is the third cytoplasmic loop that will confer the specificity. So in the upper left-hand portion of the slide, I have highlighted the third cytoplasmic loop right there, and this would represent the G protein coupled receptor that binds to the stimulatory G protein. And then on the upper right side of the slide, this would represent a G protein coupled receptor that binds to the inhibitory G protein. And what's really interesting is that now you can use the recombinant DNA technology to swap the domains, swap the C3 domain, for example, between these two receptors. Now, a cell surface receptor that would normally bind to the stimulatory G protein and activate adenylyl cyclase will now inhibit adenylyl cyclase. And then the opposite is true as well. A cell surface receptor that would normally inhibit adenylyl cyclase will in fact stimulate adenylyl cyclase.
This concludes part 1 of the cell signaling discussion.